Savoring the Symphony. The Molecular Basis of Taste Perception. 1. Introduction. Culinary alchemy. Taste perception is a sensory symphony that transforms mundane ingredients into delightful culinary experiences. At its core, the molecular basis of taste perception unveils a complex interplay of receptors, signaling pathways, and neural processing that transpires within the intricate landscape of the taste buds. 2. Taste receptors. 5 distinct tastes. The human palate discerns 5 primary tastes: sweet, sour, salty, bitter, and umami. Each taste is associated with specific taste receptors, proteins embedded in the membranes of taste bud cells. Sweet receptors. Sweet taste receptors, primarily triggered by sugars and certain artificial sweeteners, are composed of proteins like T1R2 and T1R3. Activation of these receptors signals the brain that a food item is sweet. Umami receptors. Umami, the savory taste found in foods like meat and mushrooms, is detected by receptors containing proteins such as T1R1 and T1R3. These receptors respond to the amino acid glutamate. Bitter receptors. Bitter taste receptors, a defense mechanism against potentially toxic substances, involve a diverse family of proteins known as TUSH2 rupees. Each TAS2R receptor responds to specific bitter compounds. Sour receptors. Sour taste is elicited by the presence of acidic substances. While the specific sour taste receptor is yet to be identified, proton channels, like OTOP1, are implicated in sour perception. Salty receptors. Salt taste is detected by ion channels, primarily ENaC, epithelial sodium channel, which respond to the presence of sodium ions. 3. Taste signal transduction. Molecular dialogue. Taste receptors initiate a molecular dialogue upon encountering specific taste molecules. This dialogue triggers a cascade of events within taste cells, leading to the generation of electrical signals. Intracellular signaling pathways. Taste receptors utilize various intracellular signaling pathways, including second messengers like CAMP, cyclic AMP, and IP3, inositol trisphosphate, to transmit signals to the cell's interior. Ion channel activation. In the case of salty and sour tastes, ion channels directly respond to the presence of specific ions, initiating changes in the cell's membrane potential. 4. Taste bud organization. Taste bud anatomy. Taste buds, clusters of specialized cells, are distributed on the tongue and other oral surfaces. Each taste bud contains different types of taste cells, allowing for the detection of multiple taste qualities. Taste cell lifespan. Taste cells have a relatively short lifespan, typically lasting a few days to weeks. Continuous turnover ensures the renewal of taste bud functionality. 5. Neural processing of taste. Cranial nerves. Taste information is conveyed to the brain via cranial nerves, primarily the facial nerve, 7, glossopharyngeal nerve, X, and vagus nerve, X. These nerves transmit signals from taste buds to specific brain regions involved in taste perception. Brain processing. The primary gustatory cortex in the brain processes taste signals, integrating them with other sensory inputs to create the overall perception of flavor. 6. Individual differences in taste perception. Genetic variability. Genetic factors contribute to individual differences in taste perception. Variations in taste receptor genes can influence a person's sensitivity to specific tastes, explaining preferences and aversions. Age and experience. Taste preferences can evolve with age and experience. Children may be more sensitive to certain tastes, while exposure to diverse flavors can influence taste preferences over time. 7. Supertasters and non-tasters. Taste sensitivity. Individuals fall into categories of supertasters, normal tasters, and non-tasters based on their sensitivity to bitter compounds. Supertasters have a heightened sensitivity, while non-tasters perceive bitterness less intensely. Genetic basis. Genetic variations in bitter taste receptor genes, such as TAS2R38, contribute to supertasting or non-tasting abilities. 8. Cultural and environmental influences. Cultural factors. Cultural and environmental factors significantly shape taste preferences. Cultural practices, regional cuisines, and exposure to diverse foods influence an individual's palate. Cross-cultural variability. The perception of taste can vary across cultures, impacting the desirability of certain flavors and textures. 9. Beyond primary tastes. Multisensory integration. Taste perception extends beyond the primary tastes, integrating with olfaction, smell, and other sensory inputs. This multisensory integration enhances the overall experience of flavor. Texture and temperature. Factors like texture and temperature contribute to the perceived flavor of food. The brain integrates these sensory inputs, creating a holistic flavor experience. 10. Conclusion. A symphony of sensation. The molecular basis of taste perception orchestrates a symphony of molecular interactions, neural processing, and individual variability. As taste buds dance to the tunes of specific receptors and signaling pathways, the diversity of human taste experiences unfolds. From the genetic nuances that define supertasters to the cultural influences that shape culinary preferences, taste perception invites us into a world where biology, culture, and personal experiences converge to create the delightful tapestry of flavor we savor with every bite.